Welcome to a new episode of the ITU Journal webinar series, where you can find insights and forward-looking research on future and evolving technologies. The ITU Journal is an international journal providing complete coverage of all communications and networking paradigms, free of charge for both readers and authors. This publication considers yet to be published papers addressing fundamental and applied research, building bridges between disciplines, connecting theory with application, and stimulating international dialogue. Its interdisciplinary approach reflects ITU's comprehensive field of interest and explores the convergence of ICT with other disciplines. We count on your support to make this webinar an interesting experience. Please submit your questions via the Q&A channel at the bottom of your screen. All questions from the audience will be taken during the Q&A session after the talk. The meeting is being recorded and the recording will be made available on the webinar website. Closed captioning is also available for this event. You can enable this by clicking on the closed caption icon at the bottom of your screen. We hope that you will enjoy the talk and we encourage you to stay connected until the end for the Wisdom Corner. I will now give the floor to our Master of Ceremonies.
Hello, and welcome to the new webinar series with academics of the ITU Journal on Future and Evolving Technologies. My name is Alessia Magliarditi from ITU, the United Nations Specialized Agency for Information and Communication Technologies. It is my pleasure to open our second webinar of the season with Professor Contact from Michigan State University and Stanford School of Medicine. Please submit your question via the Q&A channel. We will address them to our speaker during the Q&A session. And after the Q&A, as just announced by our avatar, please stay online. I will moderate the Wisdom Corner, live lessons. Professor Contact agreed to a personal chat. So he will share with us some lessons learned over the years that might perhaps be useful for some of you. I'm pleased to introduce Professor Ian Akilditz, Editor-in-Chief of the ITU Journal and founder and president of Truba. Professor Akilditz is Ken Byers Chair Professor in Telecommunications Emeritus at the Georgia Institute of Technology. Professor Akilditz has established many research centers around the world, including South Africa, Spain, Saudi Arabia, and Finland. He's Editor-in-Chief Emeritus of Impact Factor journals, highly cited and at the top of the most prestigious international rankings. He's visiting distinguished professor in several universities around the world. His current research interests are in 6G wireless systems, terahertz communication, metaverse, and communication in challenged environments. So Professor Akilditz, the floor is yours to present our speaker and to moderate the Q&A session.
Thank you, Alessia. Hello all. Good morning, good afternoon, and good evening. Greetings from Atlanta to our global audience. It is my honor to welcome you to the ITU Journal on Future and Evolving Technologies in short form, ITU JFET webinar series as we launch Season 4, Episode 2. This season, we are proud to present an outstanding line of speakers, and I'm delighted to introduce our distinguished second speaker, Professor Christopher Contact, today. Before doing so, allow me to provide a brief overview of our journal. Our journal was established with a clear mission to bridge the gap between academia and industry, foster collaboration, and advance cutting-edge research. Conceived in December 2019, the journal successfully published its inaugural issue in December 2020. As an open access publication, ITU Journal FET ensures that high quality research is freely accessible to all with no fees for readers or authors. Each submission undergoes a rigorous peer review process, and we prioritize timely, impactful research at the intersection of academia and industry. The success of the journal is a testament to the collaborative efforts of a dedicated team. I would like to acknowledge the invaluable contributions of Sijo Ono-san, Bilal Jamusi, Alessia Magliarditi, Erike Campilongo, Maria Otero, and many others. Their unwavering commitment and hard work have been instrumental in shaping the journal into the respected platform it is today. I encourage all of you to consider submitting your research to our journal. Additionally, if you have ideas for special issues on emerging or trending topics, please do not hesitate to reach out to us. Now it is my privilege to introduce today's distinguished speaker, Professor Christopher Contact. On behalf of the entire journal team, I extend our deepest gratitude to Professor Contact for accepting our invitation and sharing his expertise with us. Professor Contact is a trailblazer in the fields of molecular imaging, biomedical engineering, and translational research. He serves as the founding director of the Institute for Health Science and Engineering and the inaugural chair of the Department of Biomedical Engineering at Michigan State University. Prior to his current roles, he held prominent positions at Stanford University, including Associate Chief of the Division of Neonatal and Developmental Medicine, Director of the Center for Innovation in Vivo Imaging, and co-director of the Molecular Imaging Program at Stanford, in short form MIPS or MIPS, and the Child Health Research Institute. Chris Contact earned his bachelor's degree in biology and PhD in microbiology from the University of Minnesota, followed by the postdoctoral training at Stanford University. He joined then Stanford faculty in 1995, where he held professorships in pediatrics, radiology, bioengineering, and microbiology and immunology. A pioneer in molecular imaging, Dr. Contact has developed innovative imaging techniques that reveal cellular and molecular processes in living subjects, guiding the development of novel therapies. His research spans cancer biology, immunology, infectious diseases, stem cell biology, and advanced microscopy. He has led numerous NIH, DoD, and NSF funded projects and has mentored countless graduate and postdoctoral scholars, fostering diversity and excellence in academic research. Professor Contact's leadership extends globally. He is a past president of the World Molecular Imaging Society and the Society for Molecular Imaging, and of which he is a founding member. His contributions have earned him prestigious accolades, including the Achievement Award from SMI and the Britain Chance Award from SPIE. In addition to his academic achievements, Dr. Contact is a visionary entrepreneur. He founded Xenogen Corporation, I hope I pronounced it right, now part of PerkinElmer, BioEclipse, a cancer therapy company, and ExoForce, a company development exoskeletons for athletic, medical, and military applications. With an H-index of 78 and over 33,000 citations, his work has had a profound impact on the scientific community. On a personal and professional note, I had the privilege of collaborating with Chris on the Coronal project, Cooperating Cells for Human Health Through Internet of Bionanothings, from 2015 to 2020. This multidisciplinary initiative involved partnerships with the University of Oslo, Rikshospital, and Norwegian Institute of Science and Technology. It is worth noting that the project's title, Coronal, coincidentally shared a resemblance to the term corona, only to term please, which Chris and I had discussed at the time, referencing the well-known family of viruses. Unfortunately, a few years later, the COVID-19, the corona pandemic emerged, and the similarity in names led to baseless accusations from certain individuals who falsely claimed our project was connected to the virus. Their unfounded claims have been deeply troubling, but we remain focused on moving forward. In October 2024, I visited them at Michigan State University to deliver a distinguished seminar on prediction and avoidance on infectious diseases through the Internet of Bionanothings. Not only did I thoroughly enjoy my visiting all his people, like tens of them, almost 100 people, but we also engaged in fascinating discussions about research during the day and touched on politics over dinner. Throughout our collaboration, Chris's brilliance, creativity, and collaborative spirit made the experience exceptionally rewarding. Beyond his impressive professional achievements, he is a remarkable individual, intellectually stimulating, kind, and a true pleasure to work with and to spend time with. We are truly honored to have Professor Contact join the esteemed roster of speakers in our webinar series, further enriching its legacy. Without further ado, please join me when welcoming Professor Chris Contact as he takes the stage to deliver what promises to be an enlightening and inspiring presentation. Again, thanks, Chris.
Thank you, Professor Achilles. It's too bad they didn't associate Corona with the Corona beer. That would have been a lot easier to explain than the coronavirus. So thank you so much for the kind introduction, and thank you, ITU, for the invitation to present our work today. I'm going to talk about some of the recent work we've been doing in trying to build and intercellular communication networks. And we're doing this through engineering synthetic organelles and the communication networks between organelles within a cell and the cell that hosts these organelles. And this is a challenging concept even for biologists because organelles don't generally have their own networks. They have a communication pathway that largely circulates or revolves around the energy generation system of the cell. But there are communication networks that we can engineer into those systems. So this is a picture of our Institute for Quantitative Health Science and Engineering. And as you can see, the sky is always blue and the grass is always green in Michigan. So it's just a beautiful place to work. We have a tremendous group of faculty that have joined the Institute. It's only been around for about 8 years. And so 45 faculty working across a variety of different areas. Every division within the institute is comprised of faculty from multiple departments, multiple colleges, and it truly is integrative sciences. And I'm fortunate to work among a group of really outstanding scientific thought leaders. We've recruited scientists from around the world, around the country, to really work on this idea that we can develop the tools, technologies, and knowledge that will ensure 100 years of healthy living for all people. And that is what we work toward in everything that we do. So as it was mentioned in the introduction, that we developed imaging tools some time ago, and this is an example of one of them. It allows us to watch biology in living animals. And when I started in my studies in biology, we always thought of animal models as a black box where you inject your infectious disease, your cancer, your immune cell, your small molecule, and then you wait, and then at some time you guess at when to look at the animal and guess at which organs to look at. What you're seeing in this little movie is a study where we transferred DNA to the liver of this animal and watched how that DNA molecule is expressed over time because it encoded for an enzyme that produces light. These data points over here are taken every 2 hours for 2 days and every 4 hours for another 4 days to give you a sense of how a gene is expressed after it's been transferred to the liver of a mouse. This is a relatively old movie. I like to show it for a number of reasons, most of which are listed on the slide because it really creates new opportunities to watch biology and disease. I also like to show it because a single undergraduate in my lab lived in the lab for a week with an alarm clock, getting up every 2 hours and taking an image, and then we stack these images up together. The movie is pretty old. That undergraduate now is a practicing physician in Los Angeles. His name is Chris Gruber. But this movie was set up to try to demonstrate that we can watch how a gene is expressed. I can tell you what this gene did 2 hours ago, 4 hours ago, 8 hours ago. So we improve our statistics by having those built-in internal controls. We can use image guidance to sample the correct tissue at the correct time so that we're not working in the dark, but working with some guidance. And we can track labeled therapeutics. We can track the targets or both. And you'll see me jump between tracking the agent versus tracking the target as I describe some of the studies about engineering organelles for in vivo control of cell and tissue biology. We can use this to guide our omic analysis. So omics refers to the idea that we can look at large numbers of genes or proteins or metabolites. And if we can guide that with where to look, when to look, we can get more exquisite studies of our animal models. These same reporter genes work in cell culture as they do in mice. And so we can link the in vivo to ex vivo or an in vivo to in vitro studies to better refine our analysis of mammalian biology. So this is how this particular imaging system works. We take enzymes from glow-in-the-dark organisms like the firefly, and this is the 3D structure of the firefly luciferase. In the presence of energy, a substrate looks like this, and oxygen, this enzyme produces light. So we can see the abdomen of the insect glow. And so we took the gene that encodes this enzyme and we put it in every cell of the mouse. And we did this for a variety of different purposes, but one of them was to watch how small molecules like this one are distributed throughout the body. So if you take a solution of this small molecule, D-luciferin, think of it as a model drug where we can ask, where does this small molecule go in the body? So we can inject it into the peritoneal cavity of this mouse, and we see that wherever that small molecule goes, we get light. So we can watch it in the abdominal cavity, but it spreads throughout the animal. And this movie is sped up. The peak time of light emission is 15 minutes. We can compare that then to this other movie where the small molecule is injected into intravenously into the blood system, and it again goes everywhere. And in this dorsal view, you can see that it may be excluded from the brain, and we've been studying that a little bit as well. So we can watch pharmacokinetics in real time and use this as a model drug. And for that reason, we began to modify this molecule and ask, can we watch its delivery in vivo? One of the modifications we made was by replacing this oxygen with a nitrogen. Now this looks like a biomolecule called an amino acid with our carboxy and amino terminus so that we can integrate this or build it into peptides and watch where those peptides deliver small molecules. We can also link it through a disulfide bond so that we can use a targeting molecule to carry this drug in the body and watch where it goes. So this tool has enabled us to watch biology, but now we can think about how can we use this tool to demonstrate that we can control biology, and that's what I'd really like to talk about today. I've been saying that for the last 30 years, we've been watching biology, but the challenge now is to control it. So if we think about how we're going to control biology, we need to send in a signal into the body, and that's been done in a field called optogenetics, where light is used to operate optically sensitive proteins as ion channels to turn neurons on and off. The field of optogenetics is used to change animal behavior. You can make animals fearful, you can make them brave, and you can do this with optical signals into the brain. You could also turn these you can also deliver chemical signals, ultrasonic signals. You can deliver thermal energy. I'm going to talk a little bit about magnetothermal energy because with magnets, we can penetrate anywhere in the body. And with magnetic particle imaging, we can guide that magnetothermal energy to the right place at the right time. And there are other ways, of course, other signals you can deliver into the body. But to do that, you have to have some sort of a receiver of that information that can take that signal and then transduce it into some other outcome in the cell. These transceivers could be dyes, they could be proteins. For example, in the optogenetics, it is a calcium channel in the neuron that gets opened and closed with light. It could be particles or nanostructures. This is an example from Mikhail Shapiro's work at Caltech, where he takes gas bubbles that can react with ultrasonic signals to create heat and to change biology. What I'm going to talk about today is the use of organelles as engineered uh, uh, biologically encoded control modules that can receive signals of these types and then transduce that signal to a variety of different genetic networks. And so that's what we'll focus on today. The target we've been thinking about is how do we turn genes on and off? So we've been thinking about genetic circuitry, but you could also work with proteins. You could change protein pathways, and as another target for these various transceivers. And then you want to ask, what do I want to change? What can I change? And what should I not change? And some of the cell functions I'll talk about today is turning immune cells into cells that recognize tumors and kill them. We can change cellular fates and functions so that our idea here is that we could deliver a signal to a cell, tell it to become a stem cell, reprogram it, and then tell it to differentiate into a different cell type, with the idea that we could inject something in the body, have it go to the damaged heart, flip a genetic switch, make those cells in the heart into stem cells, flip another genetic switch and turn those stem cells into cardiomyocytes and guide tissue regeneration from outside the body using magnetothermal energy. We're not there yet. I wish I was giving you the talk saying, look at how we've rebuilt the heart. But what I'll show you today is that each step along the way is possible. And we're at the point now we're trying to integrate these various systems in order to do this. So like I said, our focus has been on organelles with the idea that we could engineer them, put them into animals, deliver a magnetothermal signal. And to do this, we use bacteria that biomineralize iron and make strings of magnets. They're called magnetotactic bacteria. Initially, we were developing them as living contrast, but now they can actually receive, we've been developing them as transceivers where they can receive a magnetothermal signal, transduce that into a genetic signal, and send that out to a cell so that we can then flip on a light switch in the cell and watch the whole process over time. So this is the general concept, and I'll tell you about how we're working at each step along the way, from the signals that are inputted, the signals that are biological networks within this cell that then communicate with cells that they live in. So this idea began quite a long time ago for me, and it arose from hearing Mark Ellisman from San Diego talk about using super resolution microscopy to look at organelles in cells. I didn't find one of Mark's old movies because that was some time ago that I heard him speak at a conference we call Imaging in 2020. But I was able to find a similar movie from Stephan Jacobs where the green are the organelles or the mitochondria in a yeast cell. And this demonstrates a couple of things. One, these mitochondria are networks within the cell. They aren't these little cigar-shaped oval structures that you typically see in typical textbooks. And when a cell divides, which is what you're seeing here, there are mechanisms for pulling these mitochondria into the daughter cells so that every cell has the mitochondria. So if we wanted to engineer endosymbionts, and we call them endosymbionts because these mitochondria are derived from symbiotic bacteria living inside the cell, and I'll talk about that in just a moment. But if we wanted to mimic that, we'd be able to build control modules into this cell that then get transmitted to every daughter cell. And we think of them as symbionts because the mitochondria provides energy to the cell and the cell provides proteins to the mitochondria. So they live in a harmonious symbiotic relationship. So as I was thinking about this, I then met Caleb Bell. He started a company called Bell Biosystems and was developing what he called a magnel, or a magnetic organelle, taking these magnetotactic bacteria and using them as cell tracking agents. In discussing this with Caleb, we came up with the idea that not only could we track them with the magnetic particles, but we could use that to control them. And I would propose that engineering mitochondria is a new frontier for imaging and cellular control. And when I think about this, I really like the quote by Richard Feynman where he said, "What I cannot create, I do not understand." And nobody's been able to build a mitochondria. And so what that says is we don't really understand what they are. And that's our goal, is to rebuild a synthetic mitochondria. To do that, we have to understand how mitochondria started in the first place. So about 2 billion years ago, one cell crawled into another cell, and the cell that was entering the other cell was a parasite that was crawling into the cell to steal its ATP. And it used an enzyme called ATP-ADP translocase to pull the ATP out of the host cell into itself to live off that other cell. Over time, this ATP-ADP translocase flipped around and began to pump ATP out of the cell into the host cell, and that formed this symbiotic relationship where the mitochondria provided energy to the cell and the cell provided nutrients to the mitochondria. And they originated from this obligate intracellular pathogen or parasite that evolved from a free-living bacteria. So if you think about rebuilding a mitochondria, you'd think that why don't I take a mammalian cell and an otherwise free-living bacterium and enable that free-living bacterium to get into the cell and exert effects on that host cell. So even though mitochondria have their own genome, they're obligate symbionts. They have to live inside another cell, and they don't really have autonomy for replication outside of the cell. So to engineer a mitochondria is you'd have to do it inside of the cell. So our choice was to make a otherwise free-living bacteria into a synthetic organelle or synthetic mitochondria. And again, these are somewhat difficult concepts even for biologists. And so I'm going to go through this in a stepwise fashion. So the mitochondria are naturally inside cells. They develop there as an endosymbiont, that is a parasite that lived inside the cell. And it evolved over 2 billion years to be a mitochondria and no longer has that autonomy that it had before. But we can take similarly sized bacteria, and we should be able to engineer them to enter into the cell, and they could be taken up either by the cell eating them, process we call phagocytosis, or we can coat them with cationic lipids and melt them into the cell. And we can derive a lot of lessons from other intracellular bacteria in nature to do this. So once inside, then we have to begin to think about what are we going to allow that or enable that endosymbiont to do. And we refer to them as endosymbionts, not just engineered organelles, because at this point, they're not quite an organelle and they're not quite a free-living bacterium. So we're kind of in this place between an autonomously growing organism that can substitute or coexist with a cell as an endosymbiont. So we engineer these bacteria then to make a protein shown here in the red dot. And proteins that we use, one of them is called Listeria lysine O. And when bacteria go inside mammalian cells don't really appreciate having a bacterium in their cytoplasm, so they try to wall it off and protect the cell from this bacterium. This Listeria lysine O allows it to break out of that vacuole. And then we can flip this different protein on, and that protein can be engineered with a secretion sequence. So this protein inside this bacterium gets secreted into the cytoplasm of the host cell. We can then add additional sequences to that protein so that we direct that protein from the cytoplasm into the nucleus where the DNA sits. And we can use the fact that this protein sits on the DNA and turns on genetic circuitry so that we can flip a genetic switch and convert this cell into a different phenotype. We can drive these genetic networks to give the cell additional functions, or we can actually change their entire fate and take a cell that would be a blood cell, turn it into a stem cell, and then into a heart cell or a liver cell. And that's the goal. To do this, we have to do one other step, and that is to engineer codependence. We want to make this cell dependent on this cell, and the host cell dependent on the internal cell. So that is the goal, and I'm going to talk to you about it as how we've accomplished many of these steps. But this serves as both a guide or an outline for what I'm going to say, but also a summary of where we are in our current design of these synthetic control modules that live inside cells. So if we can engineer biological remote control by recapitulating eukaryotic evolution, where mitochondria evolved, and if we could engineer these organelles from otherwise free-living bacteria to develop this mutualistic relationship, what would we do? When I first talked to students about this, they would say, would it take us 1.5 billion years to develop just like it did for evolution? And then would we need another 2.7 billion years to refine it like it took evolution that long? And my answer is, I hope not, because I'm not sure we're going to make it that long. But one redeeming feature is we can build on the 4 billion years of prokaryotic evolution, where they have evolved a whole variety of different mechanisms to interface with eukaryotic cells. And we can learn from these prokaryotes, and then we could use these synthetic or engineered organelles as biologically encoded remote control modules to direct biology. And this would give us transient control that would preserve the genome. One of the problems with many of these genetic tools is they change the genome permanently, and that is a problem. But if we did this in a transient relationship between the endosymbiont and the host cell, we could redirect the biology and then eliminate the control module when it's no longer needed. So if you had such a system, a remote control module capable of directing biology, what would you do with it? Here's one example from recent advances where they actually directly engineered organelles. I've been talking mostly about mitochondria, but chloroplasts are also organelles that evolved from bacteria. And this company, BioGlow, directly engineered those chloroplasts to express a luciferase, much like I showed you in the mice. And these plants then that have the engineered organelles just glow in the dark. And they would do this forever as long as they're growing in your home, or if you have them on your in your new remake of an Avatar movie, these plants will glow as long as they're alive. And what that tells us is the chloroplast retains enough of the ancient bacterial genetic pathways that you can introduce bacterial genes into the chloroplast and have them expressed. And for plant biologists, they probably say, well, sure, of course, we knew we could do that. But for those of us who don't study plants, it seemed like kind of a leap. The problem with engineering the organelles directly, oops, sorry, I need to go back. The problem with engineering the organelles directly is that they're not capable of autonomous replication. So in order to make plants glow in the dark, they had to get the DNA into the plant cell and that DNA into the chloroplast. And so they did it right inside the cell itself. The recent developments of genetic modifications and mitochondrial DNA make it possible that you could actually engineer the mitochondria using various nucleases and base editors. And it is being developed as a way to treat mitochondrial diseases, nicely reviewed a couple of years ago. But our biggest problem with doing this as a biologically encoded remote control module is that once you do that, that's a permanent change. That mitochondria or that chloroplast is changed forever. You can never reverse it. So if we develop synthetic organelles, then we would enable these synthetic organelles to be removed after they're no longer useful. And this would make a better therapeutic than a permanent change to the genetics of the host cell. So to create synthetic organelles, we ask, can we learn from nature? What can it teach us? And there's a number of limitations, and I've highlighted some of them to create organelles, synthetic organelles from otherwise free-living bacteria, and then using their control functions from a distance, because I think we need to be able to do this from outside the body and control them inside the body. So the problems we have are that mammalian cells don't really tolerate bacteria, but we can learn from intracellular bacteria how they do. Bacteria generally don't enter mammalian cells, but we can use phagocytosis or natural entry proteins or chemical delivery. Bacteria have many more genes and functions than are needed. So what I'll tell you about is we're developing minimal genome chassis bacteria. The genome of a mitochondria only contains 37 genes, whereas the genome of a bacterium contains thousands of genes. But we can minimize the genome, and I'll talk a little bit about that. It's difficult to maintain intracellular bacteria inside mammalian cells, but we can try to mimic some of the mitochondrial functions so that they are retained. And it will be necessary to control gene expression in the engineered organelle. And what I'll tell you about is how we're doing that chemically and how we're doing it with magnetothermal energy. And then, of course, once the engineered organelles are no longer needed, then we need to ensure that we can get rid of them. And since they're derived from otherwise free-living bacteria, they're still sensitive to antibiotics, we can build into them suicide genes, or we can develop magnetothermal destruction protocols where we can just essentially burn out the endosymbiont after it's no longer needed and leave the cell to continue with its own fates and function. About the time we were doing this, there's a great paper published by Peter Schultz's group where he put E. coli bacteria into yeast cells. This looks a lot like the yeast cell I showed you in the beginning with the mitochondria. But they've knocked out some genes in the yeast and they've added some function in the E. coli to try to create a symbiotic relationship. Again, they're working towards understanding the evolution of mitochondria and doing this with E. coli. And this partnership between the E. coli and the yeast persisted for several generations. It was a really exciting demonstration of being able to create engineered endosymbionts or engineered mitochondria. So that study used E. coli as a chassis organism, that is a chassis that we can continue to build on, that we could then create a number of different functions. But we don't think that the E. coli is the best chassis organism. It's a gram-negative bacterium that has lipopolysaccharide in its coat. Lipopolysaccharide is one of the nastiest toxins that we deal with as humans and other mammalian systems. So I'm going to tell you about a couple of chassis organisms we're working with, and one that we've been working with some time is Bacillus subtilis. And Bacillus subtilis is being worked on by Ahmed Zaria in the lab, and he's been working on it as a way to engineer neuronal cells to treat diseases like Parkinson's. What we like about this bacterium is that the genome is entirely sequenced and it's annotated. It's got a really well-developed genetic system, so it's easy to manipulate. It's got a great transporter system, so we can use that to transport signals out of the cell. It's generally non-pathogenic, and there's a relative of this organism called BCG. This is the mycobacterium vaccine that's used to vaccinate for tuberculosis. It's not used in the US, but in Europe and in Russia, they use BCG as a vaccine against tuberculosis. In this country, we actually use it as a treatment for bladder cancer. If you put BCG into the bladder, it gets taken up into the cancer, into the tumors, and creates an immune response. So there's already an example of using a related organism in a bacteriotherapy approach. So if we create an endosymbiont, this serves as an example of how that could be built on. This bacterium has a flagella, but it doesn't have LPS, so it doesn't have that toxin. It's used in industry to make a variety of different proteins using this TAT transport system, and it's a common soil organism. And like I said, Ahmed's been working with it for some time to develop it as an endosymbiont that would live inside the cell of a mammalian cell. One of the other chassis organisms we're working with is a minimal genome bacterium. Not only is this the smallest living organism on the planet, it's all entirely synthetic. So John Glass and his colleagues at the Venter Institute made a genome on a DNA synthesizer, transferred it into the Mycoplasma mycoides, and then destroyed the genome that was already there. So, so this is a bacterium that has a completely synthetic genome. It has its genome reduced to 531 kilobases, which is 473 genes. At the time they published this paper, 149 of those had no known function, but they were determined through their experiments to be essential. So they call this bug synthetic bacterium 3.0, and we use synthetic bacterium 3b. And Roxy Riddle has been advancing this approach, using this as the chassis organism. One of the problems we have with this particular chassis, and it's based on the fact that it was derived from Mycoplasma mycoides, is that mammalian cells don't even see it when you co-culture these. And so we've been trying to develop it with surface modifications to get it into cells. The other host organism that we've been developing as a chassis is Listeria monocytogenes. This bacterium is an obligate intracellular pathogen where it gets into a cell. It uses Listeria lysino, as I mentioned earlier, to get out of these vacuoles, and then it grows inside the cytoplasm of a cell and even can spread to other cells. So Satya in my lab has been working with this in partnership with Jonathan Hardy, who's also a colleague of ours at Michigan State. And as an intracellular pathogen, it grows inside cells. Here you see those red listeria, and they form little comet tails by making proteins that propel it inside the cell, but also can propel it using these actin myosin filaments to drive it into other cells. So in this case, we're taking what is a pathogen and trying to disable it. And so we're using a strain of bacteria from Dan Portnoy, where there are 4 essential genes eliminated, making it an obligate intracellular organism unable to transmit from cell to cell. And in the case of Roxy's work, we're taking a minimal genome bacterium and tried to add things to it rather than just take things out. So in my lab, we had studied Listeria for a very long time. This is work by Jonathan Hardy, where he showed that it persists in the gallbladder. It can colonize the bone. And we use, in this case, we use this bacterium that we've put the bacterial luciferase into so it glows in the dark, and we use the light from that bacterium to localize it over time. When it's in the gallbladder, we could get it to exit the gallbladder by treating with cholecystokinin, causing the gallbladder to contract and shooting the bacteria then into the intestinal tract. One last bacterium that we've been working with is this magnetotactic bacteria called Magnetospirillum magneticum. And it makes these nanomagnets in a linear structure, and they can be isolated. They make the bacteria magnetic, so you can pull them to the side of a vessel. And we did some early work using them as contrast agents with Caleb Bell and Brian Rutt, who I introduced in the beginning of my talk. And Sam Gambier had also been looking at them as ways of imaging with MRI. Here you see they accumulate in tumors using MRI to do that. We actually loaded up mammalian cells with these bacteria, injected them into the brain of mice, and we can see 100 cells or 1,000 cells that have been loaded with the magnetotactic bacteria using the effect of these nanomagnets on water to give you a loss of signal in MRI. So back to each step in this process of getting the bacteria into a cell, and then the bacteria out of the endocytic compartment. There's a lot of ways that this has been accomplished already in nature. So imagine that this is the purple bacteria that we've now introduced into the cell. It goes into the endosome and then escapes the endosome using LLO, much like I had talked about with before. Very similar to what mitochondria do when they undergo the autophagy step. So this protein, Listeria lysine O, allows the bacteria to get into the cell and then escape the vacuole. It happens in Mycobacterium. It happens with Listeria lysine O. Brucella, another bacterium, also does something similar so that it can live inside the cytoplasm of cells. So we've taken this protein and put it into the bacillus, and we've used it in the minimal genome bacteria. This protein is very interesting. It's got a unique structure that allows the ions to float out. It creates a hole inside the membrane, intracellular membrane inside the cell, and that allows the pH change to occur by these pH-dependent amino acids creating this pore structure. So now that I've kind of gone over the kind of the methods of how we would do this, what are we going to target first? And a number of years ago, Yamanaka and co-workers won the Nobel Prize for determining that these signals could take any adult cell and turn it into an induced pluripotent stem cell. And then you could put additional signals into those cells to turn those stem cells into anything you'd like, a skin cell, a cell of the pancreas, a lung cell, a liver cell, a heart cell. So we know the signals, we know the communication pathways, we know what they can do. And now the question is, could we put these into endosymbionts to drive this from inside the cell rather than using it from outside the cell? Typically, iPSCs are used to grow organoids in culture, where you take adult tissues, you take a biopsy, and you then turn them into induced pluripotent stem cells, and then drive those stem cells in culture to beating heart cells or to firing neurons. So all of this has already been worked out for us. We need to just transfer this step into an endosymbiont in order to do that. What I like about using this approach for guiding stem cell differentiation is nicely demonstrated in this study we did some years ago, where we showed that we could take one stem cell that's of the cells that make blood in our bodies. We could take one of those that was labeled with this glow-in-the-dark gene, the luciferase. We could put it into a mouse, and by 12 days, we could see approximately 1,000 of the cells that are daughter cells from the one cell we put in. And we knew it was one cell because we could look in the dish and we could see one green cell, pick it out, and inject it into the mouse. But what's really spectacular about this experiment is that one cell grew into 1,000 cells and then grew into 10 million cells. So we're assuming that our engineered endosymbionts will be a relatively inefficient process. But what this tells me is I just need to do it once, and I have the ability then of building all of the blood cells in the body from a single cell. To do this, we had to ablate all the natural stem cells in the bone marrow using X-ray, and we also had to transfer a few unlabeled cells so these animals wouldn't die of hematopoietic failure. But this demonstrates the tremendous replicative capacity of stem cells. So if we target those cells, we should be able to engineer them as they go. So let's test the basic concept. Can we set these things up? So we used LLO in Bacillus subtilis, and if LLO is left off, we don't see many bacteria inside the cell. If we flip LLO on, we see the little pink bacteria inside the cells. And we use a marker for the endosome stained in red because the endosome destroys these bacteria. What LLO does is allows the bacteria to get around the endosome and live in the cytoplasm. And you can see that here, where the pink bacteria are living in the cytoplasm away from the red endosomes in these cells. And so you can see these individual bacteria inside the cytoplasm looking much like an engineered endosymbiont. Additionally, not only do they go into the cytoplasm, but you can look at an individual living cell that has 3 of these engineered endosymbionts, and over time, you can see that those endosymbionts grow in the cell. So if you remember that movie I showed you in the second slide, where the mitochondria are throughout the cell cytoplasm, and then as the cell divides, it would pull those mitochondria into the daughter cells. We're hoping the same thing would happen here. The macrophages tolerate these very well, these endosymbionts very well, so we're not killing the host cell. So about the time we were doing these studies, there was another group that published this really interesting paper where a bacterium, Pseudomonas aeruginosa, was engineered to inject things into cells. Bacteria have these nano syringes, these molecular syringes that can inject proteins and inject nucleic acids into cells. And this is one of those secretion systems where they engineer this bug to inject transcription factors that, much as I described for our studies, then would go to the nucleus, bind to DNA, and turn things on. And they demonstrated with this approach that they could use extracellular bacteria to deliver a signal to make heart cells from stem cells. So to take this the next step, to put the bacteria inside the cell, we had to engineer it to secrete these proteins. And so we do that through the TAT system. What I like about this system is it takes a folded protein and puts it into the cytoplasm. And we put onto those proteins the ability then to go from inside the cytoplasm to inside the nucleus. And I'll show you a little bit more about that in a moment. In addition, we now have to control the expression of these genes, and we do this using our chemical control. We use sugars such as mannose that are not utilized by mammalian cells, although it is taken up. So when the mannose goes inside the bacterium, it binds to an activator, which binds to the promoter and turns a gene on. This is what we did in the study to demonstrate that we could get a large protein like beta-galactosidase from inside the endosymbiont into the cytoplasm. And this is the data from that study. If you don't add mannose, you don't turn the gene on, and you don't see it to any great extent. You add the mannose, and you see over time that you can actually deliver the protein made by the bacterium in the cytoplasm to the nucleus of the cell, and you see with induction of that nuclear localization signal that all the nuclei stain red, and that's quantified here. So unfortunately, I have to teach you a little bit about bacterial gene expression. And I apologize. I'm going to get into a little bit into the weeds here, but what's the important point to make here is that bacterial genes can express multiple proteins, in this case, 5 different proteins from a single gene driven by a single promoter. So I can put a genetic switch here that turns 5 things on, and that allows me to take all of those transcription factors and bundle them together with a singular controlling element. So we've made this cassette of transcription factors that would make stem cells. We've made a cassette that would make heart cells. We've made a cassette that would make liver cells, and we're working on making sets that would make pancreatic beta cells or skeletal cells or neurons. And we put them all into these constructs that could be controlled by a single promoter. We can also demonstrate that if we put the luciferase into these engineered endosymbionts, we can actually localize them to the liver, and they eventually accumulate in this tumor of a tumor-bearing animals. So we decided we should start to work on the ability to use these to change the immune cells to create anti-tumor responses. To do that, we wanted to develop more tools for watching these engineered endosymbionts in real time. And so rather than MRI, we've been using magnetic particle imaging, which rather than looking at the effect of iron oxide on water, like you would in MRI, it actually looks at the magnetic particle itself. And this is work by Ashley Macklin in my laboratory, where she's delivering magnetic particles to a tumor, and you can image where those magnetic particles are in the tumor on top of a CT scan of the mouse. So this gives us one more handle on how to look at and watch magnetic particles inside of a living animal over time. We can correlate those signals then, where the iron oxide is, to the signals of where the glowing tumors are, and show that these iron oxide particles actually go to the tumor, with the idea then that we can deliver an alternating magnetic field to that tumor and flip genetic switches on and off in the tumor, in the tumor cells where the bacteria reside. I'm going to skip this because it looks like I'm running out of time a little bit. So we've created a couple of constructs that actually make either pro-inflammatory responses or anti-inflammatory responses. And this is work by Cody Madsen in the lab and Ashley Makala and Emily Greason. They've done really a great job showing that if they put these engineered endosymbionts that drive the M1 signal into macrophage, they make these, they reprogram these cells into a different phenotype. And I'm gonna skip all the details in here, but the controls are extensive, and we're turning this phenotype on with this sugar mannose. You can also show that they make different when you can turn on genetic pathways in those cells by having the bacterium there itself or expressing factors that drive an inflammatory phenotype, and that's what's shown here. So with these endosymbionts that are going into macrophages and flipping genetic switches on that create a pro-inflammatory phenotype. We can do the complete analysis and show that the cells are dramatically changed when you compare the untreated to the two different treatment protocols, one with these transcription factors we call KG and another one with SK. So these are all the same cells, but being reprogrammed with those intracellular endosymbionts living in the tumor. What that does is if we just look at this data, and I'm going to zoom on that and zoom into that on the next slide, we can look at these endosymbionts going to the tumor. They are in the tumor once it's excised. The tumor can be localized with magnetic particles in the mouse or in the tumor once it's excised. And if we treat with these pro-inflammatory endosymbionts, the tumor doesn't grow, whereas the untreated tumor does grow. We've begun to look at ways to do this in the magnetotactic bacteria, but first we had to improve these bacteria. So naturally, they make these linear strings of nanomagnets. We could change them to make clumps of nanomagnets so that they now have an increased magnetic particle signature. That allowed us to track them in the body so we can watch where those magnetic bacteria are, in this case in the liver. They also express the luminescent gene, so they glow in the dark. This technique is more sensitive when the signal's near the surface, so we can see it in this approach, this imaging approach, but not this one. And then as the bacteria are cleared from the animal and concentrated in the liver, the signal's too deep to get an optical signal out, but you can still see it with a magnetic signal. Those magnetic signals would allow us to deliver magnetothermal energy to turn switches on and off in the body. So we worked with Magnetic Insight to build an alternating magnetic field generator so we could flip these things on and off with magnetic fields. The way that works is this protein binds to the promoter and turns it off. When you warm it up with magnetothermal energy, that protein comes off and the ribosome is allowed to bind. That ribosome then makes the protein. One problem we have with this genetic switch is that it quickly reanneals and flips the gene off, so we need better control there. But using this approach, we could show that we could create a magnetically regulated light switch where the more time and temperature you deliver, the brighter the genetic switch. So we not only have an on-off switch, we actually have a rheostat where the signal increases with time and temperature. We've also done this chemically. Satya in the lab has taken this construct, which is a synthetic transcription factor. It has a sequence that gets it, that allows that transcription factor to come out of the endosymbiont, and then another signal that takes it from the cytoplasm into the nucleus, where we've engineered this receiver to receive multiple signals and flip the light switch on. So we're asking this engineered endosymbiont to go into a cell and flip on a light switch inside the cell. And this is the first time this data has ever been shown, first time ever an engineered endosymbiont has been able to switch on a light switch in a cell. And here you see it in cells in culture with a temporal analysis. And this is beautiful work by Satya. And Satya then put this into a mouse where the light switch is in the mouse and the activator is in the bacterium. You inject the bacterium, it goes into the mouse and flips on a genetic switch to turn the lights on, in this case in the lymph nodes, intestine and liver and lungs, and we can watch that over time. So the first time in human history, we've shown now that we can take endosymbionts into a mouse and flip on genetic cassettes inside a living animal in the tissues of that animal. So this is my summary, and I'm going to try to skip over a little bit so that I can thank the people in my group. I've also added to my talk a few short notes we've published recently in Open Access Government. There, we describe a lot of the studies that I showed you today in these online free available articles. I have a great group of scientists that work with me. I've tried to highlight them as we go through, and I've shown the ones that contributed to the work today in red. They're really an outstanding group of investigators. Many of these students have gone on to their own careers. And in 2024, we have a new set of investigators. And I've talked a little bit about Satya, Roxy, Ahmed's work. We had a visiting scientist, Daisuke, from Japan, who's done a lot of work with us on using this for genome editing. And with that, I'll stop and take any questions. Thank you all very much.
Thank you, Chris. Very nice talk. I enjoyed it. And of course, very interesting topics. And I am trying to get questions, but anyhow, I will start the questions. By the way, I have to mention that we have a handful of people at these webinars, but as mentioned before, we put our webinars on the internet, and based on the statistics, we get hundreds and thousands of hits for the on YouTube. So so please don't be disappointed, but that's the usual number. But people are always using these YouTube or our channel, our link to access these talks. So you mentioned about engineering mitochondria, which is really very interesting topic, and I would love to work on that. But mostly you were based on the prokaryotic, like the bacteria cases. Did you or do you work on this eukaryotic, like human cell cases, trying to engineer these mitochondria, or is there any work going on the engineering mitochondria for the eukaryotic?
Yes. Yes, I showed a slide where I talked a little bit about that, and there's a nice review article in 2022. We decided to make synthetic mitochondria rather than work on the natural ones. And the reason for that is that if you work on the natural one, that's a change that's going to be there forever, for the lifetime of the cell, the lifetime of the human. So our goal is to take, since mitochondria evolved from prokaryotes 2 billion years ago, we want to recapitulate that and then make the bacteria into synthetic mitochondria. So we like that approach because it gives us much greater flexibility. And I'm noticing in the chat window that York Earwaker noticed that, of course, there are a lot of other things you could do with engineered endosymbionts, and he worries about the unintended consequences or dual use. And I agree, that's a huge question. We can use this for amazing good. We think we can rebuild liver, we can rebuild heart, we can rebuild brain, but the worst case scenario is now we're creating endosymbiotic prokaryotes that could do other things as well. So we're building into our constructs kill switches and interdependence such that if you don't maintain these organisms under certain conditions, they won't survive, and they can't cause, without understanding what those programs are, they won't cause the unintended consequences that York is referring to. So we do try to control that for obvious reasons, because this is an incredibly powerful new technology to control genetic networks inside a living animal or living human. And we do have some concerns about making sure that we protect and use these for good, not evil, and protect against the secondary uses that would be harmful.
Yeah, so York has a lot of comments, but you already
I jumped
some
ahead.
of his concerns, but that's one of the group people that you know, you look at the bottle from the negative side, right? Is empty or full.
Yeah.
You know, this is a
Well,
usual problem. I always tell this story that in 1998 we were trying to put the Wi-Fi on in our department on our floor, and my secretary came and said, "I'm pregnant and I will sue Georgia Tech if my baby will have damage from the Wi-Fi." Because there was this, you know, negative, you know, like rumor that it causes cancer, whatever the, you know, it affects the babies. And later on, this was 1998 and then 98, and then
yeah.
later, like 2004, five, then when these 3G systems were started, actually more 4G, and then there was this huge rumor going on, especially in Europe, that the cell phones are creating brain tumors. And a lot of research was done and money was wasted to find out that is it true. So it looks like we have a lot of people with brain tumor walking around, right? So billions of people are using cell phones. So anyhow, so each time you do, you know, this type of revolutionary research, then people always look at the problems from the negative side.
Yeah.
Don't
A, a
think,
good a good analogy in biology is when we wanted to get genes into non-dividing cells, we started to engineer HIV as a viral vector to carry genes into resident cells. And I have to say that there was a lot of uh, concern about engineered HIV as a gene transfer tool. But now it's used everywhere. Everybody uses HIV-based vectors to carry genes into cells, and they're called lentiviruses, these
yeah, lentivirus.
lentiviruses. And
Yes.
lentiviruses get into non-dividing cells like neurons, and I guess so does herpes simplex virus gets into neurons, and so they're used as gene transfer tools. And there was a time when the NIH didn't allow you to clone these things, and now we use them as a routine tool. So there are good examples in the field of biology, in addition to what you're referring to with the field of wireless communications, where uh, people are afraid of the unknown, and powerful technologies are powerful for a reason and can do powerful things. And it just takes some thoughtful consideration and control so that these negative uses don't get aren't possible.
What do you think about these organelles? Like, you know, it started a long time ago with artificial heart, and then you know, there is also a lot of research going on trying to create artificial liver and other organs. What is your opinion that how far are we in that research front?
Yeah.
Are these heart like artificial hearts really surviving long time, or as well as like artificial livers? What is your opinion about that?
Yeah, so so I showed a movie of a organoid, a heart organoid beat.
Organoid, yeah, organoid.
Yeah.
That's the word.
So
Yeah.
we've been doing quite a bit of work with our colleague Aitor Aguirre. Aitor developed a technique for self-assembling heart organoids where they actually form the chambers of the heart, and they can be a millimeter or two in size. So he's modeling the first several days of cardiac development using these heart organoids. And we've been looking at the communication among the cells in those heart organoids and asking how that intracellular communication helps orchestrate the formation of a heart. So the challenges there are several fold. You can make these cells differentiate into the cardiomyocytes, but we haven't really come up with a great way to vascularize that heart to get the nutrients to those cells that's necessary so they can grow bigger than a couple of millimeters. So the way to tackle that is with 3D bioprinting. And so you could print parts of the heart as a scaffold, stitch it together and let it grow in. And there are people that are doing that work. The other approach that I find really attractive is this idea that you could take an embryonic stem cell from a one species and put it into a developing embryo of another species. And in that recipient species have the genes knocked out so it can't make a pancreas. And then what happens is the pancreas does grow from that embryo, but it's the pancreas from the donor species. So you can put a rat ES cell into a mouse embryo and generate a mouse with a rat pancreas. But the rat pancreas isn't twice the size as the mouse pancreas, it's mouse size. So that tells us that there are local signals that tell those cells how to become a mouse pancreas and not a rat pancreas. So I think the idea of guided tissue regeneration is possible, and that's where what we're working toward with our endosymbionts, because we think we can guide those regenerative processes in the context of the liver or the context of the heart, and have the signals that are there already helping guide those cells to becoming heart of the right size, shape, and function, or liver of the right size, shape, and function. So those are our goals. And we can grow them in a dish now. We can begin to bioprint them. But I like the idea of in vivo guided tissue regeneration using engineered organelles as the signaling mechanism.
I would like to thank you. That's very nice. I would like to ask you also about the state of the art. I also worked on this genetically engineered E. coli bacteria cases, and there are many research results in that domain. As today also you mentioned many of them. What is the state of the art in the genetically engineered human cells? You mentioned about lentivirus, and so is there a huge progress in that domain? Also about these ethical issues of genetically engineered human cells. Where are we in research in that?
So CRISPR is now state of the art and used in every lab to edit genomes. And CRISPR can be fairly specific, and better CRISPR enzymes are being created, better guide RNAs to guide that CRISPR molecule to the right genetic sequence. Tremendous capabilities to engineer mammalian genomes. I don't usually think about engineering human genomes because I think that's a challenge, but we can do that in human iPSCs. So we do knock genes in and knock genes out of our human iPSCs, and we make heart organoids with knockouts in them to ask how those genes relate to, or how those genes can change cardiac development. We can do that in any tissue. All the tools are there, and I think that's another case where the unintended consequences could be extreme. So we limit our studies to cells and culture, and even though we do genetically engineer mice all the time, so similar techniques could be used in humans. And of course, there's a company that's been formed to try to recreate the mammoth genome and de-extinct mammoths. So that gene editing capability is there to actually do pretty substantial genetic engineering in mammalian systems.
Wow, that's interesting. Okay, thank you, Chris. Very nice, enlightening talk. And I'm sure it really helped this audience, but also in the future, there will be many others who will be interested in this talk. And I think there are no other questions. So I ask Alessia to take over and move on with life lessons. Thank you again.
You.
Fantastic.
Thank you so much,
Thanks,
Ian.
Ian.
Thank you very much, Professor Contant, for this very interesting presentation. Now we move to the Wisdom Corner. So which aims at offering a unique and special angle to this webinar series, adding a personal touch. So I would start with my first question. Looking back on your journey, what is the most valuable life lessons or even a failure you have encountered that shaped your path? How can this lesson inspire those people attending the webinar?
There are obviously many failures throughout our careers, but I'm gonna talk a little bit about more of a success or more of a life learning lesson. When I was growing up, my dad had his veterinary practice attached to the house. And my dad was this quintessential curious individual. And um, behind the house was the operating room and the kennels and the laboratory. And I lived out there with my dad, just asking questions. And we were hatching turtle eggs, we were blowing glass, we had a chemistry set. So my entire career was spent essentially asking the question, how did this happen and what does it teach me? And then when I started with my children studying wilderness awareness survival skills, I realized that one of the key questions there is asking, how did this happen and what does it teach me? So when I mentor graduate students, I ask them as they're looking at their data, look at the data and ask, how did this happen and what does it teach me? And let the biology teach us what's going on in the world around us, whether we're observing how the climate is changing or whether we're observing how a cell that we engineered responds to the signals. Continually asking that question and take it as deep as you possibly can. What that does is it leads you down a very diverse set of scientific pathways. So I'm trained as a microbiologist. My appointment's in an engineering department. We've built little tiny confocal microscopes to reach in the body. We've come up with other imaging tools. And I think if you if you if you think about the experiment and ask about the what's the next step and what can I learn from this data, it takes you down really interesting pathways, and allowing the data to lead you down those pathways makes for an interesting career and exciting career that it's just fun to jump out of bed in the morning and say, I get to go to work today because I'm going to watch for the very first time in human history an engineered organelle flip on a genetic switch in a mammalian cell. And just that statement alone, imagine being able to say that out loud 5 years ago. People would have asked, that's not possible. So yeah, how did this happen? What does it teach me?
Excellent. And in your opinion, which areas of study in the field of health science and engineering hold the most potential for future researchers? I mean, are there any emerging trends or groundbreaking topics that excite you and that you would strongly recommend young researchers to explore?
Oh, I've been asked how come I run such a diverse research program in my own laboratory? And my answer is always, I've never seen an interesting scientific question I could walk away from.
Mm.
So I don't know if I can guide someone down a path because I think there are so many amazing, interesting things to do. I would turn it around and I would say, find your passion and dive as deeply into that passion as you possibly can. And I like what Richard Feynman said is that everything's interesting if you look deeply enough. And I wouldn't pick a topic, but I would go for a really in-depth understanding of how this works and then how it can be used for good and how it can be engineered and manipulated. So I can't think, I mean, there's so many interesting things out there. I don't even know where to start. We should all be children in candy stores looking at all the possible opportunities. So what I would advise is people read and think broadly, but then focus on a specific area that tests some of the larger hypotheses. And there's a saying that we can understand the whole by understanding the part. So focus your research on a part and relate that then to the whole. And always do your studies over a range of resolutions where you zoom in to the nucleotide, the molecule, the single entity, and then zoom out and say, how does it fit into the organism, the world, and the environment? And continually zooming in and out, imaging or thinking over that range of scales as you go through every experiment is what makes life interesting. And everything's interesting if you look deeply enough, because there's always something there that nobody yet has understood or uncovered.
Absolutely, very clear. I think you have answered kind of the third question of what key advice would you give to young professionals aspiring to build a career in academia specifically and research? In your
Yeah.
experience, there's any essential skills and qualities that contribute most to the success in these fields?
So there probably was a time in human history when the known butted up against the unknown. But what I'm finding now is that one field, the known in one field, if you cross that line into the unknown in that field, you usually bump into another field. So the growth plates of our fields are bumping into other fields. And if you don't speak the language of both fields of physics and chemistry or biology and chemistry, you don't realize that you've crossed into a new area. So there's very little space in that knowledge space where you're not bumping into another field. There's probably a few little avenues that go into the unknown, but for the most part, once you bump that other field and you start looking, you realize that 5, 10, or 20 other people are working on something similar, but from the physics perspective or the chemistry perspective. And we come, in my case, from the biology perspective. So if we don't understand and speak the language of those other scientists and we get stuck in our jargon, we're not going to be able to advance our fields. And so we need to build those bridges and cross into those other disciplines. And when I had the opportunity of setting up a new institute, I always say about our institute, if we didn't need walls to hold the roof up, we wouldn't have any walls in the building because everybody works together across disciplines. In the division that my lab is in of chemical biology, we have people from chemistry, chemical engineering, but also biomedical engineering, microbiology, small animal sciences, and vet med, all working together in one group. And every large lab, and there's 8 large labs in the building, is a thematic area. And each one is like that, designed like that, crossing departments and colleges. And we have representation of 6 different colleges and 18 different departments in our one building alone. And every faculty
Wow.
member has appointments in multiple different departments. And everybody has home departments bringing students from all over the university. It's a great nexus of integrated science that is just an exciting place to work. And the reason I did come to Michigan State is to actually create that and try and realize that kind of an environment where everything crosses into different disciplines and there's no barriers or walls to scientific discovery.
Wonderful. Excellent example, very rich and variety of disciplines. Wonderful. But we're curious to know what first inspired your passion for your field, and how did you turn this interest into an important career?
Yeah, I think the best example I have is the imaging tool I talked about, where we took genes that encode luciferases and put them in different places. And uh, when I was a kid, we would catch fireflies and watch the fireflies glow. And then when my children were interested in this, we actually caught some fireflies and I put them into a plastic jar with a plastic lid. And I could see the light coming through the plastic lid of that jar. And I thought, we've been observing this phenomenon for thousands of years. And now, only 30 years ago, did we actually use it as a reporter to study gene expression in a living animal. And so I think we looked at that and we said, this is interesting. And then we said, look at all the applications of imaging this way. And then we wrote a patent on this topic, but nobody licensed the patent. So the 3 inventors got together and said, well, I guess we have to start a company, but none of us had the experience. And I was a postdoc at the time. So we started the company Xenogen. And I think when you take something that has tremendous potential, you saw the spark, you then kind of ignited that, and then helped guide the potential through all the different steps. So the first thing we did with in vivo bioluminescence imaging was to look at infectious agents. And then I said, well, this would be useful in cancer, and then it'll be useful in stem cells. It'll be useful in gene expression. And I made a conscious effort in my lab to look at all those different application areas. And now, 30 years later, in vivo imaging using bioluminescent reporters is used in stem cells, gene expression, gene editing, all these different areas. It really didn't, it wasn't enabling technology. So asking, what is the potential of what I'm doing today, and how can I bring it to the point where that full potential is realized? And it can be, it is definitely challenging and definitely takes considerable effort, but it's just fun to look back on this 30 years. The patent was written in 95 on in vivo bioluminescence imaging. So this is the 30-year anniversary of that tool. And we've been using it in everything that we study all along the way. And it's just been fun to watch.
Oh, wonderful. And my last question, among your amazing career achievements, is there one in particular, one contribution in particular, whether professional or personal, that has had a profound impact on you or others? What makes this accomplishment especially meaningful to you?
That they were done as a part of a really integrated team. The students I've had in my lab, the postdocs I've had in my lab, have just been outstanding. And it's just fun to watch people go through the thought process and come up with new and interesting ideas. And some of the funnest ones are when the student says to me, I want to do this, and I advise them against it and say, don't do that. It's going to be too much effort. And then they do it and it works and creates an entirely new area that I didn't think would lead anywhere. Friend of mine has the saying on the bottom of the email is that scientific progress is made when young scientists do what old scientists say isn't possible. And I love that. And I think that's been the funnest part of my career, is to watch what young, inquisitive, curious minds do, given an environment where they can excel.
Wonderful. Thank you so much. That was my last question. It was very interesting Wisdom Corner. Thank you for being generous, sharing your experience. Thanks, Ian, for coming back with
Thank
us.
you. Thank you again for everything, Alessia, for hosting this live lesson session. Thanks a lot, Chris. I hope we'll see each other again
I
somewhere
hope so
soon.
too. Thank you all very much.
Greetings to your wife. We had that lively discussion, remember, about that? Yeah, also my other friends there, like John and my student Bige and others. I really enjoyed my trip that time.
You inspired tremendous discussion during your visit to our institute. Thank you very much, everybody.
Thank you. Really,
all right.
Thank you. Thank you so much. We'll see you again, Ian, online on the 1st of April with Professor Melodia.
Yes, thank you. Very
thank
good.
you all. Thanks.
Thanks a
Bye
lot
bye.
again.
Bye, everybody.